Adesso?
Ok, grazie.
Ciao. Prof, una domanda. Ma la cistocitosi e la degranulazione sono due eventi comunque distinti e accadono uno dietro, cioè uno a seguito all'altro oppure in certi precisi momenti?
Allora, quando si degranulano muoiono, quindi è l'evento finale. E quindi tutto ciò che può essere fatto viene fatto prima, perché la degranulazione loro non hanno meccanismi per difendersi. È talmente forte la reazione che inducono che li ho definiti con quel modo buffo, effetto kamikaze, perché loro arrivano e si suicidano, cioè rilasciano questi granuli che contengono tutti questi enzimi particolarmente attivi che digeriscono l'ambiente, digeriscono anche loro stessi e creano un danno alle cellule tutto intorno. Quindi dove c'è un'infiammazione, come magari avete anche visto, basta solo una pustola schiacciata, eccetera, si forma un'area di necrosi da cui può venire fuori un po' di pus, un po' di cose. Il pus sono tutti tessuti uccisi, morti, tutto intorno. Quindi la reazione infiammatoria è violentissima, localizzata magari, però estremamente violenta e crea un ambiente talmente inospitale per i microbi, ma anche per i tessuti normali. Quindi è qualcosa per cui è normale che appena si è riusciti si cerchi di limitare l'infiammazione, perché sembra quasi la risposta infiammatoria importantissima, ma allo stesso tempo si va sempre in farmacia a comprare un antistaminico, un antinfiammatorio, perché si vuole ridurre al minimo questa infiammazione. Perché crea un danno, ritornando a questo, crea un danno. Quindi i neutrofili, quando arrivano, muoiono e si suicidano, uccidono tutto ciò che è intorno. Niente di drammatico, però creano delle lesioni abbastanza evidenti.
Grazie.
Allora, io vorrei lasciarvi di volta in volta le lezioni che ho già fatto, quelle del giorno prima, perché poi io me le rivedo la sera, le rimetto un po' a posto ancora, eccetera. E invece possiamo farlo questo?
Sì.
Quindi...
O facciamo una cartella e lei le lascia direttamente nella cartella del desktop ogni giorno, noi ce la prendiamo poi.
Sì, quella è la cosa più semplice. Va bene, poi facciamo
quello,
così.
Sì.
Allora, io ho chiesto sopra se potessimo fare lezione sabato, se ci lasciano in qualche modo aprire i locali, tu magari saresti capace anche ad attivare, quindi vedere questo in maniera che poi abbiamo domenica e lunedì liberi. Io sono qui se volete consultarmi, possiamo trovarci da qualche parte se avete delle domande, e lunedì fare l'esame e martedì fare l'esame. Se non è possibile, pazienza, e facciamo la lezione martedì e l'esame mercoledì.
Io la connetta l'ho portata.
Se ieri erano bloccati con PRR e CPR.
No, invece no,
C'è un po' difficile pensare di capire che non va benissimo. Come caso, secondo me.
Okay, can we start again? So, good morning. Do you have some questions about yesterday lessons? Something you want to see more in detail? If you're thinking about, in the meantime, I wanted to emphasize something which I think yesterday I did not emphasize in the correct way. And the most important receptor expressed by the cell of innate immunity, so the cell we discussed yesterday, neutrophil, eosinophil, basophil, macrophages, dendritic cell, epithelial cell, endothelial cells, are the expression of what we call pattern recognition receptor, PRR, which is a large family of different receptors, each one recognizing with very good specificity different kinds of signals which are commonly associated with microbial infection. So we have a receptor, we recognize danger signal. So the signal released by epithelial cell or by other sentinel cell after perceiving the invasion by microbes. We have a receptor, we can recognize dead cells and damaged tissue, so debris of a cell because there was a lesion on the skin or epithelial mucosa. Or the receptor which recognize some kind of molecule typical of the microbes, but shared by many microbes. So this receptor do not recognize Shigella flexneri or tuberculosis bacterium, but they do recognize something which is commonly used by microbes and we don't use. So molecules which are shared by many different kinds of microbes. And here, a receptor for nucleic acid. So yesterday, controlling this lesson, I added this slide, pattern recognition receptor, PRR. What are they? Pattern recognition receptor are receptors that interact with several molecules, evolutionary conserved and commonly associated with the invasion of microbes. So they recognize what? Danger signal, they recognize lesion of the surfaces, they recognize molecule released by microbes or molecule commonly used by microbes. Oops. Supposed to. Pattern recognition receptors are expressed mainly by cells of innate immunity, such as dendritic cell, macrophages, monocyte, granulocyte, and epithelial cell, to identify two classes of molecules. I wanted you to remember that because certainly in other courses of your training in medicine will be mentioned. This one are pathogen-associated molecular patterns, PAMP. So molecules which are commonly associated with the invasion of microbes. They, in many, in the English literature, many times they use the word pathogen instead of microbes, alluding that microbes which can induce a disease. I believe this distinction is not correct because these are associated not only with the pathogen, not only with the dangerous microbes, but with all kinds of microbes. And you have to remember and to retain that all microbes could be pathogenic if we don't have an effective immune system. There are a lot of microbes which are not pathogenic, are commensal, or are not inducing any kind of disease, only because we have a very efficient immune mechanism to control these microbes. One example typical is Pneumocystis carini. Did you heard about this microbe before? Pneumocystis carini? Never heard. Okay. This is a microbe that was absolutely irrelevant on human pathology. No one had never any kind of problem with Pneumocystis carini because it's a microbe which is apparently is not a pathogen. It's a microbe which live on us but make no any kind of disease, except when the immunodeficiencies, the acquired immunodeficiency start to spread in the population as a consequence of HIV infection. A lot of people start to die
with pneumonia induced by Pneumocystis carini. So a microbe which before never made any kind of problem, because every one of us have a lot of mechanisms to control this microbe. So this microbe was called not pathogen because was not able to make it, to induce, to cause a disease. In reality, was a microbes which is pathogen, but is only not pathogen because we control normally very well the efficiency, the diffusion of these microbes. And do you know how IDS, acquired immunodeficiency, was identified? Was identified just on epidemiological basis by the company selling drugs, because they discovered an unusual selling, consume of drugs which were supposed to control the infection by Pneumocystis carini and this kind of bacteria, which normally are not used. And then in San Francisco, in the town of San Francisco, in the area around the town, suddenly all the pharmacy wanted to have drugs to control Pneumocystis carini infection, which was something very rare and extremely uncommon. And so the control of disease control was in alarm because it was something quite unexpected. Why we have this unexpected demand of this kind of drug? And then was finally identified as this bacterium, Pneumocystis carini, which is not pathogen, was becoming pathogen because of the lack of immunity controlling this infection. But we will discuss more in detail that discussing immunodeficiency, acquired immunodeficiency. What I want you to remember that this pattern recognition receptor recognize molecule and a group of molecules, a pattern of a molecule associated commonly with a microbial infection. And I'm sorry, yeah, it was supposed to. Okay, and also with another group of molecules called DAMs, damage-associated molecular pattern. So what does it mean? When you have a lesion, we have a scar, we have any kind of wound, you have release of molecule by epithelial cell, all this kind of peptide I mentioned it initially in the first table, which collectively can be called DAMs, damage-associated molecular pattern. So all this group of molecules, which are more or less shown here, okay, here, danger, dead cell, and damaged tissue. This is a DAMP, it's a group of molecules recognized by this group of pattern recognition receptor as a DAMP molecule. Molecule associated with dead cell and damaged tissue, which in many cases are associated with microbial infection. And also this nucleic acid, they release a lot of nucleic acid by the dying cell or by the viruses or by the microbes, can also recognize. So this one are two kind of receptor which can recognize DAMP, damage-associated molecule. And the other one are pattern of invasion-associated molecule. Okay, pathogen-associated molecular pattern, PAMP and DAMP. Okay, and then probably you recall everything we discussed in this lesson. Other questions about the first? This was almost an introduction to the immune system, and an introduction in particular to the innate immunity. All the cells we discussed yesterday are the cells of innate immunity, which induce a very fast reaction a few hours after the invasion of microbes. Okay. Okay, so yesterday we introduced and we will continue discussing a few features of this very interesting complex we evolved when we become mammalian, when from fish, from frogs, antids, we become mammalian. We evolved a small chromosomal region present in all vertebrates will contain a cluster of genes defined as major histocompatibility complex. This is a complex of genes which is present, as you remember, is called in human leukocyte antigen, a wrong name, but have some reason of this name because this cell, this molecule, the product of this gene are mostly expressed on lymphocytes. All these genes are highly polymorphic, and you know very well this doesn't mean they have many alleles distributed in the population. There are many alleles for each gene, and the alleles are co-dominant. This is the second key feature of this system, to be codominant. And then the three clusters, class 1, class 2, and class 3. And we discussed it yesterday. So the cluster of this gene is very small, located on chromosome 6, all over of us. All the human beings on chromosome 6 have this cluster, about 200 loci. This is chromosome 6, and this is HLA, and these are the genes which are located on this small piece of chromosome. And class 3 genes are genes which are different function. Sometimes we think they present inside to this complex can be random, cannot have a real reasons. Some other of class 3 genes that we will see today, some of those have a role in immunity. But besides that, the two major classes of genes are called class 1 and class 2. Class 1 was the first who was identified historically by immunologists about 8 years ago, something like that. And among all the class 1 genes, 3 are the most important. Very easy, A, B, and C. And then it was supposed to be a D gene, but then slowly was understood were 3 other genes which are a little bit different, DP, DQ, and DR, which are similar, but at the same time a little bit different. So The most important, the one on which we concentrated our attention as immunologists initially, are A, B, C, and the P, the Q, and the R. And you must know that. This is a nomenclature you have to remember because it's very important. We will refer to this gene and to this region continuously in the remaining part of the course of immunology. So these are very important. You must remember the R, the Q, and the P. And you also have to remember the difference. Class 1 genes are made by a single chain. Class 2 by 2 chains, alpha and beta. And so we have 2 genes for each one, except for DR, in which we have 2 alpha, 2 beta chains, which can be used one or the other one randomly. Very simple structure of class 1 molecule. A molecule, which is a transmembrane molecule, which means it's expressed outside of the cell. The majority with 3 areas which are quite similar. We call this area domains. 3 domains which are quite similar, plus a different domain due to a different molecule with different origin, located on different chromosome, which is different also because it's mostly invariant molecule. It's equal in all the human population, almost. It's called the beta-2 microglobulin. So this molecule is a 4-domain molecule with the same structure. And the beta-2 microglobulin can be considered the initial gene, the ancestral gene, which was used by the immune system to build many other different molecules. Class 1, class 2 molecule, and you will see many other molecules. Among those, the antibody are made with this basic structure, replica of this gene. Many couple of this gene produce a very long molecule, like this one with 3 domains, or other molecule with 5, 6, or even more longer domains. So you remember that this one is about 100 amino acid, 99 amino acid, a disulfide bridge who makes this molecule a very solid globular molecule. A brick, like a brick used many times to build a different molecule by the immune system. All the molecules which use this basic structure are called molecules of the superfamily of immunoglobulins. But we will go in detail on that when we will discuss the structure of immunoglobulins. Okay, this is a globular protein. So you can imagine as a globular domain here, and 3 replica of this globular domain with some variation, one to the other one. 45 kilodaltons, highly polymorphic. The polymorphism is mostly in the alpha 1 and alpha 2 domains. So polymorphism means that the different alleles code for a different sequence of amino acids in this area. Okay. Okay, this is the basic structure of beta-2 microglobulin. And this is what can be the appearance of this molecule on the surface of all your nucleated cells. So the only cells which have absolutely no histocompatibility molecule are the red blood cell, because they have no nucleus, and so they cannot code for this molecule prolongedly. So on the surface of the cell, there are always this molecule which are different from you, me, everyone in this class have a different combination of this molecule. And this signal outside of the cell your individuality. you are unique because you can recognize as unique because the presence of the peculiarity of the combination of A, B, C, D, Q, D, P, and D, R. This is the structure ribbon design of the molecule. This one is the beta 2, the alpha 3 domain linked to the membrane, and the alpha 2 and alpha 1 outside, which make a floor of beta sheets, which is quite large over there. So, ABC, but ABC, which have a lot of different alleles. Of these many alleles, we have just two inherited, one from your father and one from our mother. But you have always to remember that these are co-dominant. So every one of us has the two chromosomes, the two sex chromosomes, one with the alleles inherited by the mother and the one with the alleles inherited by the father. So we are in generally different, quite different, both from the mother and the father, because the mother have another alleles inherited by her father or their mother and so on. So we are incompatible. You cannot transplant a skin or something from the mother. The mother cannot be a donor of a kidney, cannot be a donor of bone marrow to the child. And the child cannot do the same to the mother because in general, they are very different from one part, one set of the molecule coded by histocompatibility antigens. Is okay? So on the surface of any kind of our cells, there are these molecules which are almost floating. The membrane is fluid membrane, and on this sea of fat, these globular proteins are moving, are aggregating, and are going around on this on the surface of the cell. And we have many class 1 and class 2 molecules. So this is just a representation of class 1 A, B, C from the mother and the father, which are always co-expressed on your cells. You have to remember, and this is something to remember, that among all the cytokines we mentioned, two kinds of cytokine, interferons and tumor necrosis factor, TNF, are those which can act on the promoter of the genes coding of these genes, enhancing, increasing the amount of molecule or histocompatibility molecule expressed by the cell. And both cytokines are normally released during viral infection. So during a viral infection, it's very good for your organism to express more histocompatibility molecule. And in a few minutes, we will discuss why it's convenient to have this secretion of this cytokine, which increase the expression of this molecule by the cell around the infection. Class 2, similar structure, but more complicated because of two chains, alpha and beta chains. Similar structure, but different. And the combination with the problem with DR, in which we can, in the cell, can select it to associate the alpha chain with the beta 1 or beta 2 chain randomly. And so we have even more complication and differentiation between our individuals. This is the polymorphism of class 2. Class 2 molecules are very different from class 1 as concerns the expression. Class 1 are expressed almost by all your body cells in different amount. Class 2 are constitutively expressed, so in this moment in which you are healthy, no problem, by dendritic cell, Langerhans cell, which is nothing else than immature dendritic cell, thymic epithelial cells, and B cells. So very few immune cells do express class 2 histocompatibility molecule. And this is something you have to remember very carefully because it's very important. Various signals can induce the expression of class 2 by endothelial, epithelial, macrophages, eosinophil, fibroblast, melanoma cell. So there are a dichotomy between natural expression and induced expression of this molecule on the surface of immune cells. Okay, this is another very important point. Remember, class 1 all over, almost all over in our body. Class 2 selectively expressed by dendritic cell, thymic epithelial cell, and B cells. The 3 cell population, which are very important. So on the surface of a lymphocyte, you can imagine, let's say, a dendritic cell, you can imagine to see a class 1 and class 2 molecule all mixed together, all moving on the sea, on floating on the cell membrane. Half of them derived from the father, half of them derived from the mother. Okay, so we ended almost with this definition. This polymorphism of the molecule of histocompatibility is a way, is very important and sensitive way to identify each individual, each mammalian, which is different from the other one by the combination of class 1 and class 2 molecule expressed on the surface of the cell. Our individuality depends, is signal outside, not depend, is signal outside by the major histocompatibility context. Okay? Then we observe class 2, as I told you, they are very strange group of molecules, some linked to immunity, like complement factor, like some are cytokines, some are enzymes which absolutely have no linkage, at least so far we cannot understand any kind of relationship with that. This is the major histocompatibility complex, which is a complex which is a little bit fascinating because have not underwent the diaspora. So the gene remain associated one to the other one during the evolution. I told you, if you do that analysis on lizard, on very primitive vertebrate, to us, we see almost the same structure and the same complex of genes. By contrast, the genes in general, they move in the genome. They localize on different chromosomes, even if they are part of the same molecule. Let's say class 1 histocompatibility molecule is coded inside the complex, but the molecule can be expressed only if associated with beta-2 microglobulin, which is coded completely on a different chromosome. So it's a very strange why we have this complex, which is a complex of genes. Major histocompatibility complex is a complex of genes coding different kinds of molecules. This must be a very important message, clear message. Then we have additional class 1 molecule beside ABC in the major histocompatibility complex, we have other molecules. We will mention sometimes discussing different features, or you will discuss in different courses. Let's say in gynecology, some of that are very important for maternal fetal interaction or for the fertility. Then we have some molecules which are very similar as a structure to the one coded by histocompatibility genes located inside, but are located in different chromosomes. So this went to the diaspora, they went around, and they are very strange molecules which are involved in inflammation in various kinds of functions. But then also class 2, we have additional class 2 molecule coded inside the major histocompatibility complex. And today we will see very much in detail the function of this molecule. Now you have just to remember that there are other molecules beside DP, DQ, and ER. So HLA, now I think it's going out of your brain because it's too many times I repeat the same, are molecules signaling our biological individuality. But the major problem is how come that we maintain this complex system, the polymorphism? How come that in Locarno the people don't start to have all the same histocompatibility molecule? Because they're not a very large population, in the past was not a lot of communication. How come that progressively in the colony of the animals, which are living in colony, we don't tend to have a decrease of the polymorphism? So how come in the human population and in animal population, the polymorphism is maintained? So you understand, this is a problem of selection of a sexual partner. So how come this polymorphism is maintained in mammalians? What are your ideas? You understand? Do you realize what is the problem? The problem is we have many alleles. How come that progressively during all the year of human civilization, the alleles do not tend to be unified? Most of the alleles are lost, and we simplify, we become just to have the same alleles or very few alleles. How this polymorphism is maintained? This is quite a fascinating problem. Do you have any suggestion of that?
To be protected by several antigens.
Sorry?
To be protected from several antigens.
Yes, this is true why we want to have, but what is maintained? How come that you are probably all the gene different from him and from him? And so this, I don't know if you understand, is really a very provocative kind of problem. When you select your sexual partner, you select a person which is rich, is beautiful, handsome. You don't care about the histocompatibility antigens. This is less problem. You see a nice girl or very handsome boy, you don't, what is your HLA, what is your... So this is quite interesting. And certainly there's some way, probably, by which the HLA influence our selection of sexual partner. This must be something influencing our selection, which is absolutely, we don't know in humans, but we can see that quite clearly in animals. Okay. Now you have a problem when you will find a girl or a boy to check what is the HLA. And okay, the first evidence of that starts from this kind of experiment in which you put a male mouse here and you put a female of the same strain, a female of a different strain. Apparently, as far as we can judge, equally nice, very nice for same age. So you put one, this is white, let's say a strain black, and the same strain of this. Okay, I have to, in mice, we have mice which have a strain which have share all the histocompatibility antigens, are called inbred strain of mice. So the inbred strain of mice are obtained by crossing a male and female, brother and sister, for at least 40 generations. After that, all the genes of the strain of mice are absolutely identical. So you restricted the HLA polymorphism. You have just one polymorph kind of histocompatibility antigen. So this is a mouse which has one set of histocompatibility antigens, and you put one female here with the same histocompatibility antigens, and another here with different histocompatibility antigens. And you see how many times this mouse is going in this direction or in this direction. And it's not an all or nothing phenomenon, but you observe for, let's say, 200 situations, 200 times, and you see there is a preference, there is a bias, there is an influence of the male to go toward the area where you put a female which has different HLA antigens, HLA genes. So the mouse can select, can understand without seeing, because it decided without, so it was not possible to see the female, just by perceiving something is going in one direction or the other one. So this male can have an intercourse with the same female of the same histocompatibility antigens and produce a progeny. So they are completely fertile. But there is a preference to a bias to go toward a different female with different HLA. Okay? Then instead of putting the female here, you can put the sawdust of the cage in which was a female of the same strain of a different strain. And you see that probably the smell, which is the one which makes this animal to select and to go toward something which has a histocompatibility different, and probably the signal is given by odors going on the air. So it doesn't matter there is the animal here, but just the odor of this animal. It's clear, this experiment. So it suggests that their signal, they having certain kind of haplotype, or certain kind of histocompatibility genes influence our smell, which signal our odors around, and we can recognize different smell, not we, the mouse can recognize different smell and be attracted a little bit better by a female which have a smell different from this own. So why don't try to do the same in humans? And this was very complicated experiment, a called Wetkin experiment made in 1994, and several times it was repeated with different values. Very complicated. I hope you will be not disturbed by this experiment. But they take ladies were students on the course of biology and psychology, and they were asked to be careful about their nose, not to take any kind of spray, don't have any perfume and so on. And they sometimes they asked the men were students of chemistry, physics, and geography, were asked to wear a plain cotton t-shirt for 2 nights and refrain from anything that might affect the odor. Don't do sex, don't do smoking, don't do drinking alcohol, don't use deodorant, or even entering in the smelly room. Okay, so male have to wear the t-shirt for 2 days, not change, day and night. And after 2 nights, the men wore t-shirt were placed in cardboard with triangular holes. And then each woman smelled 6 different t-shirts and ranked them on a scale from unpleasant, intensity, pleasantness, and sexiness. So wash odor was a little bit exciting. It was something quite interesting that when Kenneth Cooper analyzed that for okay, the outcome was the woman ranked a t-shirt to be more pleasant and sexual when worn by men with different histocompatibility genes from themselves. This is a main message. So probably there is a smell message we deliver around us, which signal in certain way, coupled with the kind of receptor, smell receptor we have in our nose, which recently have been found coded also by genes located inside to histocompatibility complex. We deliver some kind of signal we may influence in certain way, but certainly is not all or nothing. But a general bias, very little, something you can see doing some analytical analysis, mathematical analysis, not yes or not. It's just a general preference to person who signal outside through the odor, they are different from yourself. Okay, this was even studied further by another experiment. So it may be that subconsciously we favor sexual partners, they have different HLA genes from us. But this is very absolutely minor effect, but it's an effect. And this was studied also with a strange population of American, in America. I don't know if you heard about this movie with Harrison Ford, Il Testimone in Italian. Okay.
Speaks about in the
Speak louder.
it
is about
I don't know, if policeman or something else.
No, it's Philadelphia population is called Amish.
Amish in the farmer.
Farmer. What is the peculiarity of this Amish?
That they work in the field.
They work in the field, yes, but
without you use are not modern. And
okay, this I don't know if you heard about in Pennsylvania in particular. There are some population of early immigrants from Holland in general, which are a little bit crazy, and they establish a community which maintain absolutely the rules of the time they arrived in United States. So 1700 and something. They dress in a strange way. They don't use a car, but use only horses. And they are a community, a small community, which interbreed only inside the community, except people decided to go out and escape of this community. And this was a very nice movie. This was a detective who was involved in this community and was having very strange relationship. This is called Amish and is relatively famous because of this movie of the 1985, so very old movie. But this was a famous actor, was called Harrison Ford, and I recommend this movie if you have possibility to see. It's interesting. But another community, less well known, is called Uterine. Again, also this community are crazy, similar to the other one, less famous, but doing almost the same thing. You see, they dress in very strange ways. This is a very recent picture. You can do that now. This house is a modern house, but they dress in a very strange way. And they maintain the rule of 1800, 1700 and something. Okay? And one of the rules of they live in different farms, in a big area, in different farms. And when the boy are 18, the boy, a certain period of the year, I don't know exactly, have to go to all the farm and stay one day in every place or 2 days and see the girl. And then there is a big dancing finally, a big festival, a dancing, and all the boys select the girl, or the girl select the boy. I don't know, but it was something like that, and they get married when they are 18 years old. This is the rule of the community, absolutely strange and peculiar. But this community is a community very friendly. So what easy for scientists to study, and there are many studies on uterite, because they derive from the town from Holland, it's called Uter, because they are modern, open-minded, except completely crazy, but open-minded. So they accepted to have a test by the doctor, and they do not refuse interaction with the society. Okay, this is a typical girl of this population. They dress in very traditional way and so on. So what was the story? Was done several tests. It was found that it's possible now to type all the population, all the 18 years old boy, and to know what are the histocompatibility A, B, C, DP, DQ, DR expressed by the boy. This is a routine test you can do on the blood. With a center, we can do that. And you can see what is the polymorphism of the boy, and you do the same with the polymorphism of the girl. And then you see that the probability that a boy and a girl get in love and select each other as sexual partner is not random on the basis of histocompatibility antigen. But again, there is a very evident bias, but just a bias. It's not something, I repeat, it's not something absolute, but it's a bias, just a little drive to be select one who have a different histocompatibility antigens. And here, this is just a summary of that. Gene in the major histocompatibility complex have been associated with the choice of the sexual partner and odor preference in a variety of animals. And I just summarize a few experiments of that. Also, the role of HLA genes in human mate choice, the choice of the sexual partner has been controversial. Studies in the uterine have demonstrated fewer than expected number of couples which match for HLA. So it was random, and this is a population is small, and the polymorphism inside the population is limited. In any case, the probability that a boy and girl share the same antigen, histocompatibility antigen, is lower of what is expected by just a random crossing of the population. So there is also, in this case, is a bias. Demonstrated a fewer than expected number of couples will match for HLA haplotype with the same kind of HLA, suggested that in this population there is avoidance of mate which HLA haplotype similar to the one haplotype. Then, if you want, you have this slide, you can go further and explain. Also, they study the polymorphism of the receptor, and was very difficult to have any conclusion. But this was the final message I wanted to leave you, that the polymorphism of HLA is maintained certainly in animals, and probably in certain way difficult to calibrate really, also in human population, because we can be a little bit more attracted by someone who has a different HLA from us. And this probably is linked to kind of smell a person has. And the smell of a person is not coded by HLA, absolutely, but HLA coding immune system mostly. So certain bacteria which are living on me are not living so well on you, because you have different kind of histocompatibility antigen, different level of complement, different level of different factor. And you have a smell which is different from mine. So probably the HLA influence our general smell, affecting the population of bacteria which are normally living on us. And this can be perceived, and the same for you, by the girl and by the man. So there is a mixture of this combination. I wanted just to say that because this is an interesting evolution of the histocompatibility engine, and say histocompatibility engine are doing signaling our individuality, but probably influencing the maintenance of the polymorphism of the system. So now we move to the today most important part, which is the continuation of that. There is a high number of alleles of loci coding class 1 and class 2 molecule. The alleles that each of us inherited from the mother and the father define our biological identity. But we see that class 1 as class 2 molecule also present on the cell surface fragment of protein we call peptide, either coded or endocyted by the cell. This peptide presentation forms an important communication code leading to T cell activation. And I will spend the next hour trying to explain to you as best as I can what does it mean, this point B. So the histocompatibility molecules signal outside our peculiarity. But but they also so, first of all, what a peptide is. What?
A peptide. A sequence of amino acids.
Sorry?
A sequence of amino acids.
Yes.
Particular amino acids.
It's a fragment of a protein. How long is a peptide?
10,
How many
15,
amino acids?
10, 15.
10, 15, 20. If it's going longer, it's not a peptide, it's just a protein. Protein is 100 to 100 amino acids. So it's a fragment of a protein. Peptide is about 20, 15, 8 amino acids, the one we will consider. If you see from the top the histocompatibility antigens, we can see this area where the domain alpha 1 and alpha 2 are associated. You see the two globular domains. Okay. Here, alpha 1 and alpha 2, they form like a area, a small area, a grove in English, un buco, una zona incavata. We call grove this area, where normally there are peptides inside, both or class 1 in this case, and where is one class? Okay, class 2. Also here we have this grove here. Okay, and when we go to the ribbon presentation of histocompatibility molecule, this is the beta 2, the globular domain of beta 2. The alpha 3, this is the membrane of the cell. This is outside. This is the alpha 1 and alpha 2. And this ribbon scheme shows you here there is an alpha helix like a lip of this area, where this is the floor of this area. So this is alpha 1, alpha 2 globular domains. There is a grove here where sometimes there is something like this one, orange substance, a peptide, which enter in this cavity. Enter in this cavity. So between domain alpha 1 and alpha 2 of class 1, or domain alpha 1 and beta 1, there is a cavity in which the two domains form a floor, which is normally made by beta sheet, And we have the border, which can consider like the lip of this area, which are this alpha helix here. Okay. Now, if we go back to the picture, I'm sorry, but tomorrow I will organize better this lesson. Okay, this is the alpha helix I've shown you before. One alpha helix, this is the second alpha helix, and I don't know, you can see well the cavity here, the groove of histocompatibility molecule. This is an experiment made by a very smart lady who was able to crystallize, was the first one able to crystallize the histocompatibility molecule. And she was very happy to have this design, they isolated one molecule, one kind of histocompatibility molecule of class 1. They isolated, they produce a lot of this molecule, was able to obtain a crystal, and this is a crystal images from the top of the histocompatibility molecule, human histocompatibility molecule. So real picture in reality. But she was very surprised when we published this was a paper on Nature, that inside to this growth here was always something different from the various molecules she isolated. So not always the same. Was a fragment of a protein, which is shown in red, which is not constant. Here I was able, all the structure is the same because she isolated one molecule of one kind of histocompatibility entity. So it was perfectly equal. But every time they had something, she was able to stain with a different color because it was changing continuously. And this was discovered, okay, here, these are the two alpha helix. This is the floor. This is alpha 2, alpha 1, beta 2, and alpha 3, and this is the cell membrane. Okay, so this lady, Pamela Bjorkman, was able to crystallize the histocompatibility antigen. So from this picture, obtained this drawing. This is a mathematical model called ribbon model of the beta sheet, alpha helix, and so on. So derived from this crystal. But she was also very much surprised to find that here in this cavity, like in your teeth cavity, you sometimes have a fragment of food. Here you have a fragment of peptide. And she was very surprised, and this was a major observation by Pamela Bjorkman, which published the structure of human class 1 histocompatibility antigen A2, so the kind number 2 of the histocompatibility antigen, the A, there is A1, A2, A3, A1600. She was isolating the A2. She published on Nature in 1987, and she found this kind of problem. This is a lady, Pamela Bjorkman. At the time, she was a postdoc in the laboratory where this was isolated. So Pamela Bjorkman in 87 discovers that the majority of histocompatibility molecules have a peptide inside. Like in the cavity of teeth, sometimes we have a fragment of food we have been eaten. But this is not sometimes. Always the histocompatibility molecule have something inside, have a peptide. And for a long period of time, this was thinking was just a chance, was a cavity, and the cavity, there's fragment going there. Then it was discovered that this is a very important mechanism by which we signal what is going on inside to the cell. And I will try to discuss with you this phenomenon. So far, it's clear the presence of peptide associated with class 1 and as well with class 2 histocompatibility molecule. You don't have on the surface of your cell any histocompatibility molecule without a peptide inside, probably. Or very, very few are without peptide. There are some, but only few. Okay? So if we make the same drawing we have made before, this is a surface of any kind of your cell on which the histocompatibility molecules are floating, you can see, you can put in every one a peptide, a fragment, which remain associated with the cavity. More or bigger or the same or different fragment can associate it. So we don't have just the histocompatibility molecule, but we have histocompatibility molecule expressing, and we call presenting, showing outside some fragment, some protein fragment. Okay? Okay. So, if you have here the design of class 1 molecule and here the class 2, you can consider that always there is a peptide which is not the same bound to the groove or the cavity which is formed from domain alpha 1 and alpha 2 or alpha 1 and beta 1. Okay? And here is a typical representation. You will see many times this drawing, which is taken directly from the crystallographic study of Pamela Bjorkman. You can see the peptide, the fragment of a protein, here as the lips, if you want, of this cavity, the alpha helix, and here is a floor made by beta sheet where this peptide is located. The alpha helix of the alpha 1 domain, the alpha helix of the beta 2, and this kind of structure. Again, I put it just back, not at the beginning, but here you can see the peptide, which apparently is put here, which can establish some relationship with other part of the area. So the new information is, okay, histocompatibility molecules, but always with a fragment of protein associated with the molecule. And again, here you can see another, the alpha helix and the floor, alpha of the alpha 1 or alpha 2 domain, or alpha 1 and beta 2 domain, depending class 1 or class 2. And this is the peptide, which should establish some relationship with the amino acid which are nearby and establish certain kind of local relationship. Our class 1 antigen displayed fragment, we call peptide, of proteins localized in the growth made by domain alpha 1 and alpha 2. Okay. So, if you have a different histocompatibility molecule, you can understand that you present a different peptide. You present peptide, you cannot present because the growth of your histocompatibility antigens, the polymorphism of your histocompatibility antigen, the sequence of amino acid here you have may be different, maybe is different from the one from is sequence. So the peptide which can interact here, establishing some interaction, electrostatic interaction with the amino acid, with the shape of the cavity, can be different from you and you. So not only histocompatibility antigens signal our individuality, but histocompatibility antigens cause a different presentation of peptide. So the peptides, the repertoire, the kind of peptide you present are different from those you present or you present, because you have a different kind of histocompatibility antigen, and the difference is normally in 1, 2, 3, 10, 15 amino acids, mostly of the alpha 1 and alpha 2 domain of class 1, or the alpha 1 and beta 1 domain of class 2. So each of you presented different peptide, or at least partially different peptide. Because if you have an alanine here, and instead you have a phenylalanine of a tyrosine, you cannot put the same peptide. The shape of the growth can be very different. So if you have here different charges, this peptide cannot fit on you and you, but only in one of you, and the other one may be a peptide which is a little bit different, or the same peptide put in a different way with different kind of orientation. So going was a different presentation of peptide to T cell, and the consequence of that, the polymorphism of your histocompatibility molecule decide if you can respond or not to certain antigens. So you have a shapes of your histocompatibility molecule in which a peptide from an influenza virus cannot enter very well. You will not present this peptide, and you will have a poor response to influenza. By contrast, he can present very well, he can have a fantastic response to influenza, and so on. So every one of us, having inherited molecules which are different from the other one, have a growth which is different. And the kind of fragment, the kind of peptide can fit in the growth is different from one of us. Each of us have a different repertoire of peptide can be located inside to the cavity of alpha 1 and alpha 2 domain, or alpha 1 in beta 2 domain. Is okay? This is a really key point, so please. The question is, from where this peptide originates? And I have to say immediately that in the few cases in which the histocompatibility molecule reaches the membrane without peptide, which is relatively rare, in less than 5% of your molecule, your histocompatibility molecule, the peptide can derive from the outside. From the other one, the peptide derive from very complicated mechanism that I would be very happy to explain to you in the next half an hour. Can we interrupt for 10 minutes, 5 minutes? Okay, I've received a message by Professor Messina. You know him? He says that he will be very happy to open the. Ok. Buongiorno, lunedì 19 sarò lieto di aprire personalmente il dipartimento alle 9 per lezione del professor Forni. Un caro saluto, Salvatore Messina.
Lunedì, come abbiamo chiesto. Quindi,
Va bene?
Facciamo, volendo, con questo giorno riusciremo a finire ancora prima.
Come
Sei pazzo, compare.
Sei pazzo, ieri alle 1:30 abbiamo finito. Stamattina sono le 6.
Sai cosa è successo?
Cioè, se lei riuscisse a finire, se lei riuscirà a finire il programma in questi 5 giorni, finendo un'ora prima del pomeriggio.
Alle 5 o vuoi alle 4?
Alle 4, no, intendo prima ancora, se lei, dipende lei, non lo so. Se lei non fa niente.
Vediamo oggi come va, ecco. Solo per intenderci, io faccio domande al test solo delle cose che abbiamo discusso insieme. Se ci sono delle parti che dovete studiare da solo, vi prego, studiatele, ma non ci saranno domande su questo. Casomai non riuscissimo a finire, perché lì sul libro vi potete trovare le parti che non riusciamo. Voi avete avuto anche un testo sui vaccini? Sì o no?
Sì, sulla mail c'è. Io mail c'è, sulla lista dei libri. Sì.
Io ho più di là che di qua.
Ecco, per cui per esempio i vaccini è un argomento che possiamo discutere se ce la facciamo, se non ce la facciamo, voi avete un bel testo di riferimento che è abbastanza, l'hai letto anche? No. Vabbè, è abbastanza chiaro e semplice, quindi.
E quindi l'esame rimane per il 20, giusto, no? Se facciamo il 19 l'esame è il 20.
Il 20.
Ok.
Il caso è morto, spero che non vi crei problemi. E nello stesso tempo, se sabato e domenica avete qualcosa da dire, volete che ci troviamo un momento da qualche parte, discutere qualcosa, io sono lieto di farlo. Cosa si fa? Qui si va su in montagna a camminare?
L'unica, sì, o è prendere la funivia
E poi?
E poi su si arriva e ci sono vari percorsi per comunque scendere. E in più su c'è il belvedere, diciamo, quello sé. Oppure si fa un lago e si va alla scuola. A scuola è un bel porto, e un bel lungo lago e
e poi da qui alla scuola quanti sono? Ah, quindi un giorno vado su e un giorno vado sull'altro.
No, perché l'ho fatto
E i professori sopravvivono sempre, i vostri professori, a tutte queste ore di lezione?
C'è qualcuno che ogni tanto
fa colla.
ci hanno mandato a casa.
Perché non le paghiamo?
No,
perché
sì, è dura. Cioè è dura per noi, ma capisco che è dura anche per voi.
Perché io insegno una cosa molto strana in Vietnam, in un corso tipo questo, ma è dopo la laurea, loro la sono però molto meno preparati di voi, quindi sono laureati, e faccio una cosa analoga mattina e pomeriggio. Però vado molto più lento perché loro proprio mancano, e anche tanti film che avete visto che magari vi sono parsi un po' ingenui, la aiutano molto.
Ma lei da dove viene?
Io vengo da Torino.
non è
No, solo il primo giorno. Il primo giorno era stato poi non so.
però questi famosi gelati
Non ti mando, dobbiamo mangiare.
Buongiorno.
Ciao. Ciao.
Aspettate. Ma perché non me la comprate?
Aspetta che torniamo a sapere.
Vedi, me la dici? Questa è la tardi.
Il banco.
Ciao.
Ciao.
Posso avere anche un caffè? Come?
No, ce n'è una dentro.
No, però qui no.
Grazie.
Posso gelato lo devi provare. Lo devi provare.
Buono sto gelato.
Ci vuole proprio una crosta, cioè vabbè, una crosta di
parte blanco liscio ad esempio.
il corretto classico, solo che dentro è ricoperto dalla cioccolata della Kinder e poi gelato.
Quelli si trovano
Sopra una crosta di Kinder Bueno.
Sapere cosa sono i
No, qualcosa di tranquillo, si llama un po' di
Ah, non parlo di quella
sì, la tavola da 4 pannelli.
tutti gli elenchi, no, dovete, ma anche delle altre cose, quando vedi elenchi, tanti numeri, eccetera, cioè proprio i concetti di base.
C'è la funzione, dove vediamo elenchi, funzioni, cose.
Ogni cosa, in generale.
Di tutte queste
parts, purtroppo non c'è bisogno.
Tipo questo dei recettori mi sono imparati node, toll, node, scavenger sempre con i node.
Poi prepari uno tuo
Voi avete mica internet da mandare un messaggio al professor Messina?
Sì, sì.
Ok, se gli mandate un messaggio che lo ringrazio di cuore. Vabbè. Per lunedì.
Per Giovanni.
Oppure se no date la password al prof.
Sì, infatti.
Come?
Qua c'è il wifi, se prende la password, se
Sì,
vuole glielo può mandare anche.
Sì, lo mando poi adesso quando vado. Ma
Per il momento ringrazialo tu.
Professor Forni.
sì, professori e tutti noi, non so.
Ah, sì.
Ed è simpatico questo professore?
Sì, no, troppo simpatico.
Vabbè, se devo giocare, non lo so. No.
Cos'è un Lille?
19.
Deve stare un po'. Perché non c'è una cosa di note di mattina?
Ok, ricominciamo.
Ma quando è
Già vedi questi volte.
So the problem is from where these peptides, which are associated with the majority of histocompatibility molecule, originate. And this is very interesting to consider what is going on our cell. Probably you know better than me, but we have generally 10 to the 6, 10 million of active ribosomes, which produce almost 10 million of protein every minute in our cell. So it means that all the incorrect and surplus protein are bound to ubiquitin molecule present in the cytoplasm and are destroyed. So we continuously made a new protein and we destroy old protein. Are you familiar with this phenomenon? You already studied now we will see some detail of that which are extremely critical in understanding how these peptides are presented. But this is a key point. Every part of our body is new. It's very rare to have parts which are more than 1 month old, or very rare to have part which is older than few months. So as a contrary to a car, which is never repaired, or you just repair one piece, so we are always made new. All our proteins are new, are living for a few hours or very short period, but we continuously destroy old protein and substitute it with new one, with a very continuous process. Because of that, we need always to eat something. And if this is an old protein or an incorrect protein, or a surplus protein, protein I produce in excess, the protein is linked to the ubiquitin and become so-called ubiquitinated, and ubiquitinated protein lose the conformational structure and are sent to the proteasome. And the proteasome is a structure like this one, made by different globular structure. Each one contains hydrolytic enzyme. So this is a structure which destroys the protein. Here, there is a linkage with ubiquitin, so the protein are made to enter the proteasome, and once they enter the proteasome, they are destroyed. The caps of the proteasome bind the ubiquitinated proteins. Ubiquitin is removed and is saved to be used again and reutilized, while the protein is introduced inside the whole of the proteasome. And this is the part, the external part of the proteasome. Then there is a core, a cylinder made by 4 rings of 7 units, alpha, beta, beta, alpha, which have in general very strong ability to destroy hydrolytic activity, to destroy the protein. So if this is the protein which has been linked to ubiquitin, so ubiquitinated protein, the ubiquitinated protein is driven to the cap of the ribosome or proteasome to the ubiquitinated protein. The ubiquitinated protein is introduced inside to the proteasome, and a peptide fragment of the protein are going in the cytoplasm. And this is happening millions of times in every of your cell in every minute. So we are continually destroying and making new protein with this process. So renewing the inside of your cells. Okay? And these are peptides. But in the cytoplasm, there are a lot of enzymes. The peptide is very easy to be digested because the protein has a tridimensional conformation, makes the protein very resistant to digestion. But here, the peptides are immediately destroyed by a lot of enzymatic activity present in the cytoplasm. So this peptide have a half-life of a few minutes and after, or even less, and after that are destroyed. Okay? So the protein in this way is completely destroyed. Chopped in small pieces, the peptide, and the peptide destroyed by the enzymatic activity. The proteasome digests the protein and produce 8, 9 amino acid long peptide. And these peptides are quickly digested by aminopeptidase in the cytosol and endoplasmic reticulum. So no hope for the peptide to survive. They are destroyed as an amino acid. The amino acids are used again to build a new protein in the ribosome. It is correct, we speak well with what you know already. Yes? But a few peptides, which is the majority is destroyed here, a few peptides are able to enter the endoplasmic reticulum, where the amount of peptidase is much lower. So we call lucky, happy, fortunate peptide because are the ones which are protected, can survive a little bit longer. The other peptides are immediately destroyed by the peptidase in the cytoplasm. So there is a passage of a peptide in an endoplasmic reticulum that is where the protein are synthesized. And this passage is not random, it's not as designed here, but is made possible by this complicated structure which selectively allow to enter a few peptides only, but the lucky peptide is the one which can interact with the amino acid structure of this pump of peptide, which can allow a few peptides to enter inside to the endoplasmic reticulum. So it's a passage which is regulated by a structure which is called TAMP, transporter associated with antigen processing, TAP, which are the structure which permit to a few peptides to enter inside the endoplasmic reticulum. The lucky peptide are those able to bind particular transporter on the membrane of endoplasmic reticulum and be happy because they arrive in the endoplasm where they have less probability to be destroyed. Okay, so we say lucky, but peptides are not lucky or not lucky. Just they enter or do not enter inside to the structure. So we have this structure, you have to remember TAP, transporter associated with antigen processing, which I don't know if you remember, but we will see very shortly, is a molecule which is coded by the major histocompatibility complex. So we have some polymorphism. Not all of us have the same TAP, and in any case, the TAP is coded inside the major histocompatibility complex. So the story is like that. The peptide, the protein, ubiquitinated protein, destroyed it by the proteasome, lose a tridimensional conformation, made a peptide. The peptides are immediately destroyed by peptidases present in the cytoplasm, but a few of them, random, but because they have some feature, can interact with the TAP and be transported inside to endoplasmic reticulum. Is okay? What is going on the endoplasmic reticulum? In the endoplasmic reticulum, we have the synthesis of new protein, many new protein which are produced in the endoplasmic reticulum, and then they go to the Golgi apparatus, they become glycosylated and moves to different parts of the cell. Among the proteins which are synthesized in the endoplasmic reticulum are the new histocompatibility molecule, class 1 and class 2 histocompatibility molecule. And here you can see an initial class 1 molecule, alpha 1, alpha 2, alpha 3, which cannot stand because it's not yet associated with beta 2 microglobulin, so need a molecule which acts as chaperone, stampella, which can help to maintain the conformational structure. If not, this molecule will collapse. So this is a newborn molecule, class 1 molecule, okay, which move in the endoplasmic reticulum and then travel to go inserted to the membrane of the cell to substitute the all histocompatibility antigens. We renew our histocompatibility molecule, the glycoprotein, almost every 8 hours. This is a half-life of a histocompatibility molecule. So every 8 hours, we have new molecule, we destroy the old one, and we substitute on the same membrane with the new histocompatibility molecule. In general, we have this 8-hour interval survival of the molecule. So this one is a just synthesized molecule which arrived here. The beta-2 is independently produced. It produces coded by a genome at different chromosome. Is arrived here, and we have the assembling. This is called calnexin, but I don't want you to remember the name, but just to remember that a chaperone molecule which help to stand the molecule and help to the organization is removed when the beta-2 microglobulin is inserted, the calnexin is removed, and we have finally the formation of a new class 1 molecule with beta-2 and alpha-1, alpha-2, alpha-3 molecule. Also in this case, we need other chaperone protein. And this second one is called calreticulin. You have to know this name, but I don't want to remember for good. Just remember that we need some chaperone protein in the initial phase of assembly of histocompatibility molecule. And here we have, when this molecule is just born, is assembling, we have the peptide, what we call the lucky peptide. The few peptides we were able to interact with TAP and to be transported inside. Where we have just some peptidase, we can cut some pieces of the peptide and form various peptides, and some of those, random, by chance, can enter in the grove of histocompatibility molecules. There are many peptides different. Some have charges which cannot fit. Some have shapes we cannot fit. Among, you can think about 1,000 of these different peptides deriving from the protein which have been destroyed, and some of that random, one very well, the other not so well, can enter and fix in the growth of histocompatibility molecule, which are just made in this moment. We have also an aminopeptidase associated with antigen processing with this name, RAAP. You are not asked to remember this name, which can trim, can adapt this peptide, which cannot enter the growth, to cut a little bit and perhaps help to find a size just which can adapt to the growth. So we, in this moment, all of us, we are doing this in our ribosome, and our histocompatibility molecule of this cell are progressively associated with the peptide, have other complicated chaperone protein, tapazine and calreticulin here, and the peptide finally enter in the growth. And the peptide is very important, like pietra angolare, is something which stabilize the tridimensional structure of alpha-1, alpha-2 domains. They linked, they adapt to the peptide as they enclose the peptide in the growth. So we have the formation of this complex, the peptide, the histocompatibility molecule, and we don't need at this point the chaperone protein. And finally, we have the molecule which is ready to move and to travel to the membrane. But you have to remember that LLP1 and LP2 are TAP1 and TAP2 transport a peptide from the cytosol to endoplasmic reticulum, where they are associated with class 1 ABC, are coded, are class 2 molecule coded beside DP, DQ, DR. So we have a certain kind of polymorphism of this transporter. So your transporter are not exactly the same as the one of your friend, but are similar. They have a different kind of they can associate it with a different kind of peptide. Are between the polymorphic genes are coded by the polymorphic gene located among the class 2 histocompatibility molecule. The basic structure is not too far from a general class 2 molecule, TAP1 and TAP2. But also we have LMP1, LMP7, which are constituents of the proteasomes. So of the part which is streaming the protein, also that are coded by the histocompatibility complex. So the kind of proteasome you have is different from each of you. Some are more active, some are cutting better, some are cutting less well, certain kind of peptide. So you have to understand then the kind of peptide you generated here, the kind of peptide you generated depend on the structure of the proteasome. Certain part of the proteasome are coded by the gene located inside the histocompatibility complex, and they are polymorphic. So they are not this part equal among of you, but they are different. So the peptide, if this is an albumin, let's say an old albumin molecule, the albumin fragment he generated perhaps are not equal to the one you generated, because you have a different fragment, different polymorphism in the activity of the proteasome. So we generated different peptides, each of us. Some are similar between us, some are different because the polymorphism of some structure of the proteasome. But also the entrance inside to endoplasmic reticulum is not the same in all of us, because this structure can be a little bit different between the various of us in this room, because we have different genes, different polymorphism. Since this structure, TAP1 and TAP2, transporter of peptide to endoplasmic reticulum, are polymorphic. So they are different between the human population, being the gene localized inside the histocompatibility complex. So very complicated polymorphism. And finally, this ERAP trims the peptide, so a few of them can finally adapt to histocompatibility complex, and the interaction between the peptide and the growth is very complicated. The interaction between the peptide and the amino acid or the peptide growth of the cavity depend on multiple interactions. So it depends on the shape of the growth. Each of you in your polymorphism have a growth with a different shape. But also the shapes depend the electrical charges. Some protein, some peptide cannot at all enter in certain growths. Others can fit very well. But the same growth can bind 1,000 distinct peptides with different affinity. So the growth is there, and different peptide with a different sequence of amino acid can compete for the same growth. And this is supposed to be the peptide with different areas which interact with electrostatic forces, because of also the morphology of the growth allows or not the interaction with the peptide. And here again, the drawing you have in your book, you see here there are some electrostatic ionic interaction. This can be hydrogen bonds, but all these traits, the hydrogen bond and ionic interaction, are effective just when there is the very close spatial complementarity. So the structure of the peptide, which is this one, can fit very well with the feature of the growth. And every histocompatibility molecule has a different growth. So you can imagine that repertoire of peptide you present, and so on is quite different, not completely different, but quite different because the tap polymorphism, the proteasome polymorphism, because the polymorphism of the growth of the histocompatibility molecule. All these three events can change the peptide which can adapt to the growth. Okay? And this is a class 1 molecule with a cavity here. This is a peptide. You forget this part, and this is the presentation of peptide into the growth. So you understand now quite well that the polymorphism of HLA gene causes a different presentation of peptide. So this imaging endoplasmic reticulum with the newborn histocompatibility molecule which travel towards the membrane where they will insert. They have, the majority of them have a peptide associated with the growth. But the peptide here is different from this one, it's different from this one, or derived by different fragments of the same protein, or from the different protein which have been destroyed with a housekeeping mechanism by the cell continuously, as they travel towards the membrane, where they move progressively towards the membrane. They form post-Golgi vesicle, which travel to the membrane. Finally, the membrane of the vesicle fuse with the cell membrane, and the histocompatibility molecule, they diffuse here, and the membrane open, and the histocompatibility molecule become inserted inside, outside to the cell. And you have here and every nucleated cell of your body express 10,000 different cell peptides associated with the different A, B, C molecule, class 1 molecule, the 3 of the mother, the 3 of the father. Each one present different kind of peptide on the surface. And you do that every 8 hours, more or less. So this molecule will stay on the surface of the cell for 8 hours, then the molecule is removed, destroyed, and substituted by new molecule which arrive. So we have continuous renewal of your histocompatibility molecule and the peptide associated with them. So the histocompatibility complex signal who you are, but also you are who you are in association with a different peptide each of you is destroying in this peculiar period of the life of the cell. Every 6 hours, 8 hours, these class 1 molecules are substituted by new one bearing new peptide and so on. And this is a critical point. What does this mean, this complicated mechanism? This complicated mechanism is very important to signal. Every cell shows on the membrane, so outside of the cell, what is going on inside to the cell. If the peptides are normal, it means the cell is doing everything normal. They always boring the same peptide. But if the cell is somewhat altered, or if something is living inside to the cell, the peptide repertoire could be different. And they signal outside of the cell. So this is a key point. In this way, continuously, every 6 hours, we show on the membrane what is going on in the cytoplasm. You can imagine we are always speaking about completely black, completely dark inside of our organism, no eyes. So the cells are always touching each other, communicating each other through the histocompatibility molecule. And they do interact, controlling if the histocompatibility molecule and the peptide are the normal one of Guido Forni, of each of you, or are something altered, not normal. And so will be some kind of reaction against anomalous peptide or anomalous histocompatibility molecules. All our cells express class 1 glycoprotein and thus display peptide from the protein destroyed in the cytoplasm. Are you okay with this sentence? The polymorphism of HLA genes provide a molecular mark of uniqueness of our biological individuality. HLA class 1 molecule constantly report on the cell surface sample of all the protein that the cell is making and destroying. They reveal what is going on inside to the cell, sheltered by the cell membrane. You understand? This is the evolution of this mechanism to control the microbes who were so smart not to live in the outside of the cell, where you have a lot of peptides, you have macrophages, you have a trap given by neutrophil, you have a degranulation. So very difficult life aggregate. But try to go inside to the cell, sheltered by the membrane. I try to show that in very primitive way. Sorry. Okay, look at that. If you have a microbes who is living outside, the life of the microbes outside of the cell is very, very difficult. You have a peptide, antimicrobial peptide, you have lysozyme, you have phagocytosis, you have all the mechanism we spent the morning of yesterday discussing the afternoon, all the cellular mechanism which can attack these poor microbes, invading microbes. Then we have a complement. You will see this terrible activity of the complement. We have antibody. So a few of them, very smart in the evolution, try always to exploit our body to survive and to take the energy, invented a way to go inside to the cytoplasm. If they are going there, they cannot be detected. They are sheltered by the membrane. And for one microbes inside to the cell, they can proliferate and create a big colony and then destroys the cell and diffuse in the cytoplasm. So this is a mechanism exploited by a few bacteria, but exploited by all the viruses. All the viral infection are due to a virus which is able to enter the cell, proliferate inside to the cell, exploit the cell, produce millions of viruses, and then kill the cell and diffuse and infect another cell. This is typically diffusion of viral infection, okay? But here, if the cell proliferate, the cell produces the invading microorganism proliferate, it will produce protein, peptide envelope protein of the virus envelope. And so some of them will be displacing the peptide or deriving from the normal protein of the cell. And so some peptide will be presented outside, which derive from the catabolism destruction of the overproduction of viral protein or microbial protein. So the histocompatibility molecule will present also peptide which derive from the invading microbes. And so outside of the cell will be possible to detect what is going on inside to the cell. This is important of this mechanism. Now we go back. Okay, can we have an interval? No, take this one.
Okay,
Poi magari saranno cortissimi anche questi. Sono tre.
Class 1 major histocompatibility complex proteins display short peptides or antigens derived from normal cell proteins. The MHC complex has two subunits. The smaller subunit, beta-2 microglobulin, resembles an immunoglobulin domain. The larger alpha subunit also has an immunoglobulin-like domain, which is linked to a head domain containing the antigen binding group. The antigen binding groove in the MHC head domain is built from two walls composed of long alpha helices that rest on a floor composed of an 8-stranded beta sheet. The peptide on display fits snugly between the helices in the groove.
You see very well, the peptide can be seen some part outside.
The peptide backbone is bound at both ends by highly conserved regions of the MHC protein. Some peptide side chains extend downwards into specific binding pockets in the groove, while other peptide side chains project upwards, where they can be recognized by T cells. MHC class 1 proteins display their bound peptides on the cell surface for immune surveillance. Immune cells, called cytotoxic or killer T cells, for example, express T cell receptors that bind to the MHC head domain and the bound peptide. If the cell expressing the MHC protein displays a peptide foreign to the immune system, the T cell is activated by this receptor MHC interaction. The activated T cell then proceeds to destroy the abnormal cell. Cutaway views of this peptide-bound MHC protein complexed with a T cell receptor reveal the exquisite precision with which the interacting surfaces fit together.
Okay, I think it was very clear the interaction of the peptide with the group.
Create a stunning website. I've already done it with Wix. Let me show you how it's done. I'm going to need to put these. I can't see without them. Just go to Wix.com. There are loads.
The folding and assembly of MHC class 1 molecules takes place in the lumen of the endoplasmic reticulum. The initial folding of the class 1 heavy, or output chain, is aided by the chaperone calnexin. The partially folded chain is transferred to a second chaperone, calreticulin, which aids the further folding of the chain and the association of beta-2 microglobulin. Other proteins, ERK57 and taposin, associate with the nascent class 1 molecule, which binds to the TAP transporter via taposin to form a peptide-loading complex. The peptides that bind to the MHC class 1 molecule are generated by a large protein complex, the proteasome, which is found in the cytoplasm. The proteasome degrades proteins within the cytosol of the cell to produce short peptides, which are then transported through the endoplasmic reticulum membrane by the TAP transporter. Some peptides do not bind to the MHC molecule at all. Others can bind, but are unstable. These are released from the MHC molecule, a process called peptide editing. Finally, a peptide binds to the MHC molecule with high affinity to make a stable complex. This causes the final step in the folding of the class 1 molecule to take place, and the dissociation of the peptide-loading complex. The peptide-loaded MHC class 1 molecule is now free to exit the endoplasmic reticulum and be transported, via the Golgi apparatus, to the cell surface, where it can be recognized by the antigen receptors of CD8 T cells.
Okay. This is a case of the infection by a virus. So if we have an overproduction of viral protein, the virus producing itself, also the viral protein, surplus of viral protein are ubiquitinated, destroyed by the proteasome. They lose the conformation, they enter the proteasome, they are chopped in peptide, which are viral peptide, are not your peptide. Some of those peptides compete with your peptide to enter the group of histocompatibility molecule. They compete. You can imagine that simultaneously you have the destroyer of your protein. So they enter there, and depending, they can compete for the same growth and enter formation of association with class 1, sorry. And so they signal that something anomalous is taking place on the cell, and this can be recognized by T lymphocyte. I will discuss this afternoon. Okay, thank you. Scossa. Grazie. So this is the story of class 1 peptide. I hope it's clear to you. You have some problem? You understand that almost all your histocompatibility molecules display a peptide. A peptide which can derive normally from the normal housekeeping of the cell, which destroy continuously protein, and some peptide associated with histocompatibility molecule. And we display on the surface normal fragment of the normal protein destroyed by our cell. If we have an infection, if we have a viral infection, we have a cold, we have any kind of viral disease, we have a peptide, viral peptide compete with your peptide for the histocompatibility antigen. If the virus is reproducing itself very fast, at a certain point we have more viral peptide than your peptide. And this can be detected by T lymphocyte, which can destroy the cell. And this is a key mechanism by which we recover by viral infection. We recover by the cold because our T lymphocyte were killing the factory of virus, which will release millions of viral particles. But also, we have a peptide associated with class 2. And this is completely different story. Class 2 is not associated with the protein inside the cell. The class 1 are always associated with protein which derive from the protein destroyed in the cytoplasm. Okay? These are peptides which derive from protein outside of the cell. And I will try to explain you this mechanism, which is also very interesting. Peptide associated to class 1 derive from cytoplasmatic destruction of the protein. This is clear to you. By contrast, peptide associated to class 2 derive from endocyted protein, eaten protein, phagocyted protein, panocyted protein. Everything the cell is interacting inside. You have always to remember, and this is another key point, that only few cells of your body express class 2 histocompatibility molecule. They are expressed by dendritic cell, B lymphocyte, and few other cells, thymic epithelial cells. So normally very few cells present the peptide which are endocyted. By contrast, all your body cells almost present the peptide which are destroyed into the cytoplasm. This is very important point again. So class 2 peptide phagocyted are presented only by a few cells of the immune system. Remember that class 2, the PDQDR molecule are expressed by dendritic cell, thymic epithelial cell, and B cell. Langerhans cell is nothing else than immature dendritic cell. And it can be expressed also but during a stimulation by interferon and TNF, by endothelial, epithelial, macrophages, eosinophil, fibroblast, melanoma cell. In any cases, but constitutively or activated, there is about 10 kinds of cells we can present class 2 histocompatibility molecule and class 2 associated peptide. By contrast, class 1 are presented by all your body cells. Okay? Our class 2 fragment peptide of protein, peptide that localizes the growth might do this class 2 molecules. This is okay, here the growth of class 2 is more flexible because it's not done by one single protein, but are two different chains which are associated, so the growth here is longer and more flexible, so can adapt longer peptide in class 2. And this is a series of events taking place. Here have a peptide, a protein, which is outside, which is endocyted, phagocyted, panocyted, doesn't matter in any, all these mechanisms by which this is put inside in the vacuole of endocytosis. As I reported several times yesterday, the vacuole of endocytosis, endocytic vacuole, fused with lysosome, form a phagolysosome. When they fuse with the lysosome, is activated proteolytic enzyme. And proteolytic enzyme will start to destroy the protein which has been ingested. Okay? And then we have the formation of peptide. And if we have a normal cells which is doing this endocytosis, these vacuoles go to the membrane and release the fragment of what has been digested. We saw that yesterday in several conditions about macrophages, neutrophil, and also granulocyte. The phagocytosis, destruction, and release of peptide outside in the cytoplasm. Okay? When we have the fusion of lysosome with a vacuole of endocytosis. But in the immune cell, in dendritic cell, in B lymphocyte, and in other cells which express class 2 antigen, we have also, when we have the maturation of class 1 antigen in the endoplasmic reticulum, we have also the formation of the new class 2 histocompatibility glycoproteins, which is traveled towards the membrane. Also, class 2 are synthesized new every 6-8 hours. So you can imagine here the synthesis of alpha chain and beta chain. And we have several chaperone protein, but one chaperone protein is this one with a pink end, which is called invariant chain. And this invariant chain is a chaperone protein, very important, which go along to alpha 1 and beta 1, and with the top, which is called clip fragment, which is a pink round area, closing the growth of histocompatibility molecule, class 2. So in this postgology, the molecule is closed, the growth is closed by the part of the invariant chain. The pink fragment of the chaperone chain, which is called invariant chain, is closing the growth formed by class 1, by alpha chain and beta chain. Okay? So if the peptide here arriving through tap 1 and tap 2 in the endoplasmic reticulum, they cannot enter in the groove of class 2 molecule. Why? Because the groove is closed by the clip fragment, the pink area. You can imagine that here they are assembling of class 2, but simultaneously we have the assembling of class 1, okay? But class 1 are full with the mechanism I just described before, which is taking place also here. But here, the peptide cannot fit with class 2 glycoproteins. Why? Because the groove is closed by the pink fragment of the invariant chain. The pink fragment, which is called clip. Do you understand? So independent synthesis of the two chains of class 2 is the compatibility molecule. The invariant chain binds the two HLA chain and close the peptide growth. So synthesis of the alpha chain, synthesis of the beta chain, the invariant chaperone chains go along, keep this molecule stable, and at the same time, the fragment, pink fragment called clip fragment, close the growth. You can imagine that simultaneously here are the maturation of class 1. You have simultaneously here there are a lot of peptide transported by tap 1 and tap 2. But this peptide cannot be and cannot enter in the groove of class 2 because class 2 is closed, the groove is closed by the clip. So this is a segregation of kind of peptide which are presented by class 1 and class 2. Class 1 is linked, is bound to the peptide which enter in this in the endoplasmic reticulum. This cannot happen with class 2 because the groove is closed by the clip fragment. So endoplasmic reticulum, we have tap 1, tap 2, a lot of peptide arrive in endoplasmic reticulum when we have the synthesis of alpha chain and beta chain. They are put together through invariant chain, which close the growth and does not allow that possible peptide which derive in the cytosol by destruction of the protein, as we discussed with class 1, can enter here. They just enter in the class 1 because the class 1 have chaperone protein which do not close the growth. Okay? It's clear because this is very important selective mechanism. And what there will be the question, why class 2 do not present peptide destroying the cytoplasm? Because the growth is closed by a fragment, the pink fragment called clip, which is closing here.
But is it synthesized inside the endoplasmic reticulum?
Sorry?
Is it synthesized inside?
Yes, all is synthesized in the endoplasmic reticulum. All the protein of our body are synthesized there. They are assembled in the endoplasmic reticulum, class 1, class 2, and all the other proteins.
Also the class 2 can present the viral peptides. If, for example, there is a virus, and this virus is phagocytated into the phagolysosome.
If the virus is phagocytized, but normally it's not really. In this case, yes, it's a very mechanical event. It's not against everything is phagocytis can be presented in class 2. but normally the virus are not phagocyted but able to infect directly. Can you understand the compartmentalization? Here, the peptide derived by the transport, but the peptide cannot be bound to class 2 because the growth is closed by the clip. And so we can follow the maturation of class 2 histocompatibility model. They assemble independently, associated by the invariant chain. They clip close the growth. They are assembled, they move in post-Golgi vesicle and move towards the membrane. Okay, associated with the invariant chain. Post-Golgi vesicle move towards the same membrane where class 2 molecule will become inserted. During this journey, this vesicle fuse with phagolysosome, which is arriving from the outside. The catepsin of phagolysosome digests the invariant chain, and so the invariant chain is destroyed, remains a clip fragment closing the growth, but progressively a peptide can displace the clip fragment and associated with class 2. The clip fragment of the invariant chain, we have the digestion of invariant chain, still close the peptide growth, but the clip associated with a chaperone protein, which is another class 2 DM coded by histocompatibility complex, they do together association, and the foreign peptide, they remove the clip, the growth now is available for the foreign peptide. They are associated with the foreign peptide. So here is a phagolysosome, which have destroyed the protein which has been eaten by the cell, endocyted proteins. These are the peptide of endocyted protein, which arrive in the post-Golgi vesicle. Here we have the class 2, initial class 2 with the invariant chain, chaperone chain, which close the growth. The enzyme deriving from the proteasome, derived from the endocytic vesicle, destroy the invariant chain, remain the clip fragment. The clip fragment is removed when TM molecule go to act as a chaperone protein and allows the growth to be available to the peptide which have been digested of the phagocyted molecule. And this is a mature class 2 histocompatibility molecule which travel towards the membrane. So the event is like that: lysosomal enzyme, digestion, small peptide about 10, 80 amino acid. At this point, we have the same time the maturation of a class 2 histocompatibility molecule with a clip which is closing the growth. They feel together. The DM molecule help to remove the invariant chain, to remove the clip. The peptide deriving from the phagocyted protein, phagocyted structure, compete for the growth of class 2. And finally, class 2 insert on the membrane and present outside what the cell was eating. So something of the surrounding of the cell. And here again, the same drawing: phagocytosis, phagolysosome, peptide production, fusion together, the digestion of invariant chain, the clip still associated. This is not shown here, the DM molecule, which allows the clip removal. The peptide can associate it with class 2, and class 2 finally inserted on the cell membrane. Every 8 hours, the cell renews the class 2 histocompatibility, 6-8 hours with renewal of class 2 histocompatibility molecule. So the surface of a cell is always class 1 a class 2 molecule with different kind of peptide associated. We display the outside of our cell class 1 and class 2 peptide, which are completely different origin. So the polymorphism of HLA class 1 provides a mark of our uniqueness of our biological individuality. Class 1 molecule constantly report on the cell surface sample of all the proteins the cell is making. They reveal what is going on inside the cell, sheltered by the cell membrane. Class 2 molecule constantly report on the cell surface sample of the protein in the cell microenvironment, what the cell has been eating, has been phagocyting. They show to the T cell what is going around the cell. So very important and selective compartmentalization. And these are only presented by specialized cells, which are specialized in presenting the peptide which are deriving from the protein eaten by the cell. And the cell which express class 2 histocompatibility molecule are also called antigen-presenting cells. All our cells express class 1 glycoprotein and thus display peptide for the protein destroying the cytoplasm. All our cells. But a few lymphoid cells only express class 2 proteins. So this is a major difference. One is displayed by all. This one is displayed by just a specialized population, the so-called antigen-presenting cell. Thus, the peptide from the endocyted molecule and microbes are displayed only by a restricted array of specialized lymphoid cells, defined as antigen-presenting cell. It's okay.
Only the APC cells present the class 2 proteins. Only the antigen presenting cells present second class.
Yes, but you can put in reverse. The second class, the antigen presenting cell are the cell which express class 2 antigens.
T cells, B cells are antigen presenting cells, no?
No, T cells are not antigen presenting cells. The antigen presenting cells are dendritic cell, B cell, epithelial cells of the thymus.
Okay.
These are Langerhans cell. By contrast, they can also act of antigen presenting cell after stimulation, after induction of histocompatibility molecule. Can also be neutrophil, can be eosinophil, can be various kinds of other cells. But just after induction of the expression of histocompatibility, class 2 histocompatibility molecule. So, Again, very simple design, a surface of any kind of your cell, but this cell had class 1 here and class 2 with 2 chains. One at beta 2 and the other one at not. We signal our biological identity on the surface of our cell, expressing the peptide which normally are destroyed, are made by destroying the protein of our cell, or phagocyted from the outside of the cell by dendritic cell and other kind of cell. In addition, on the surface of our cell, we signal what is going on in the cytoplasm and what the cell has endocyted from the outside, from the surrounding of the cell. This is the normal situation by which we show outside what is going inside or what is going around. And we associated what is going around to class 2 histocompatibility molecule. So class 2 reveal what is going on inside the cell. Okay. Now, who care about the signal? Who care about the signal of our individuality? And who care about the peptide we present? This is a very complicated mechanism, very sophisticated, to signal what is going on inside to the cytoplasm or around the cell. But who care of that? And we will pass this afternoon discussing who care about that. But just to summarize it, there is a cell which is called T lymphocyte, which is obsessed by the histocompatibility molecule. T lymphocytes do not interact with any antigen. They don't see bacterial protein. They don't see any kind of foreign substance. They only care about the histocompatibility molecule.
Both class.
Both class. So the T lymphocyte, we will see this afternoon in detail, have a receptor on the membrane specialized to interact with the histocompatibility molecule. T lymphocytes do not recognize any viral protein, do not recognize any bacterial protein. They don't have pattern recognition receptor. Okay? We are completely in a different situation. T lymphocyte recognize only histocompatibility molecule of class 1 or class 2. And they are obsessed to control your histocompatibility molecule. They establish an interaction with the cell, and we will go in detail, but just to summarizing the event, they scan with their receptor the kind of class 1 and class 2 histocompatibility molecule we express, and if they found histocompatibility molecule which are not correct, or correct histocompatibility molecule associated with wrong peptide, they become activated. If not, they control the cell. They found the cell is full of very regular, boring peptide, nothing exciting. They detach and go to control another cell. This is a surveillance made by T lymphocyte constantly, or what is expressed on the surface of your cells. If they do interact with the receptor which interact both with the histocompatibility molecule and with the peptide, and if this interaction they have no particular activation, the receptor is not activated, the cell detach and go to control another cell. So this is what I explained before. And when the histocompatibility molecule present a peptide, foreign peptide, a T lymphocyte can be activated, can recognize the foreign peptide, the anomalous peptide presented in a very strange situation, and become activated. Killing by T killer cell is the most important mechanism of healing from viral diseases. All kinds of viral diseases you had in your life, rubella, mumps, cold, influenza, and so on, has been controlled, has been healing because the T lymphocyte was able to kill the infected cell before they were able to release millions of viral particles around. If the cell presents the anomalous peptide, the T cell can recognize the presence of something anomalous and induce a local exocytosis of perforin, exactly as we saw the mechanism with natural killer cell. This perforin enters the cell and destroys the cell by digesting the protein of the cell and inducing the cell to commit apoptosis or dying by necrosis. Okay, so in conclusion, this peptide presentation is a very sophisticated mechanism to control our homeostasis if we remain the same. If we change something during our life, the T lymphocyte will try to very precisely destroy the anomalous cells, presenting anomalous peptide or anomalous histocompatibility molecule. The same mechanism is activated in the rejection. When you reject a heart, you reject a kidney, depend on recognition of a foreign histocompatibility molecule or anomalous peptide. This mechanism is extremely important because it's the only mechanism by which we can control the microbes which are able to grow inside to the cytoplasm, inside, sheltered by the cell membrane. If not, we have no way to control a viral infection. So if we can see again another representation of what we saw. This is a membrane with histocompatibility molecule, which do express a peptide, and a killer T cell control if these peptides are normal or anomalous. And the peptide derived by the protein, which are being destroyed by the proteasome, the peptide which are transported in the endoplasmic reticulum, is associated with a newborn class 1 molecule and go to the membrane. Okay? But the most dangerous viral infections are made by virus with contra-evolved mechanism to resist to this powerful control. So when you will study a disease like herpes virus disease, the one who makes a fever of the lip, you have a mechanism by which the virus is able to interfere with this mechanism of control. If this mechanism of control is working, you will be healing from all kinds of diseases because it's very, very efficient. But the viruses, certain viruses, evolve a mechanism to block the peptide entrance. They inhibit the TAP working. They can inhibit the association here of the peptide. they can inhibit various kind of expression of histocompatibility molecule. You will see when you will study the various human diseases, the major disease due to the virus is a contra-evolution of mechanism to escape to this very powerful mechanism of control. Okay. The last conclusion is this one. To have certain kind of histocompatibility molecule or other kind can be very important because of the peptide you present on the membrane. So it's clear that to have a certain kind of histocompatibility molecule, you can make you better able to fight a particular infection, because they can pick up the appropriate peptide to be presented to the T cell very well. Let's say, against malaria, for instance, some of you present the peptide of malaria parasite very well. Some of you do not present very well. If the malaria is not existing in this area, both of you will live exactly well. In certain condition, will be a selection of certain kind of histocompatibility molecule. And this is probably the reason why in certain areas of the world, we have more frequency of certain histocompatibility molecule, and in other area, different kind of histocompatibility molecule. The frequency of certain kind of alleles of histocompatibility molecule, because having or not having a certain kind of growth can provide you an advantage or a disadvantage against the disease. But the diseases are very different. So in the different areas, what is an advantage in one area can be a disadvantage in another situation. So it's because of that we are extremely polymorphic. For example, having HLA B7, so B, we saw many alleles of B. Some of you will have the B7 allele. Some of you have not. If you have the B7 allele, the B27 and the B14 is a protective against the spread of HIV virus, whereas having B35 and B07 is associated with a fast progression of ADS. So with the same kind of viral infection, certain person have an advantage because presents a peptide very well and can fight against the virus very efficiently, others not. And so with many, many other diseases. So there is not a combination which is good and one which is bad. There are some combination which is good in certain situations and other which are good in other situations. You understand? So this is not a grove, a fantastic grove which is fit everything. Every grove has a repertoire of peptide which can fit in this grove and can provide an advantage in certain situations, but can be a disadvantage in other situations. Some combination of HLA genes are especially protective against particular disease, and those versions of HLA genes propagate in the population of the world area where the disease is endemic. Let's say malaria have selected around the Mediterranean area certain kind of HLA type, which are more provide a better way to fight against malaria. And so other kinds of endemic disease select in the different areas of the world a prevalence of some of this type of histocompatibility molecule. I think I've finished. Yes, then we can see this one. But
when we are saying that the HLA complex is against the spread of a virus, no? We're saying that it binds the peptides, the viral peptides, with high affinity or low affinity. So high affinity because it's quickly recognized and cell destroyed, or?
Well, I think affinity is not the real word. Can present the peptide or cannot present the peptide.
Okay.
There are certain groups which cannot present the peptide.
In this case, they
have a very strong disadvantage.
Yes, because the cell is not recognized by this.
Yes, by contrast, other people present very well. And you know very well that if we have influenza virus here, some of you will have a very dramatic disease, relatively dramatic, you also have a mild disease. Part of that is depend on the mechanism of presentation of the peptide. This is just about the mechanism of presentation of class 2.
Chains of the MHC class 2 molecule assemble in the endoplasmic reticulum, the ER, with a third chain, the invariant chain, shown here in blue. A portion of the invariant chain binds to the peptide binding groove of the MHC class 2 molecule and prevents peptides or unfolded proteins present in the ER from binding. The invariant chain also guides the transport of the class 2 molecule out of the ER and through the Golgi apparatus into a vesicle that eventually becomes part of the endocytic pathway by which pathogens and foreign proteins are taken into the cell. Progressive acidification of this endocytic vesicle activates proteases that cleave the invariant chain in two places, leaving a small peptide, the class 2 associated invariant chain peptide, or CLIP, bound to the class 2 molecule. Engulfed pathogens or their proteins are precipitated by acid-activated proteases into peptides, but these cannot immediately bind to the class 2 molecule because the CLIP peptide still occupies the peptide binding groove. The removal of the CLIP peptide is the function of a specialized MHC class 2-like molecule, HLA-DM, which is also present in these vesicles. It functions as a catalyst, coordinating both the release of the CLIP peptide from class 2 molecules and the binding of pathogen-derived peptides. The MHC class 2 peptide complex is then transported to the cell surface, where it can be recognized by the antigen receptors of CD4 T cells.
Oh. Okay, so remove this tool from here. Okay, do you have a question? Something is not clear, or you want to discuss something in particular? So in the test will be, final test will be a question. Why class 1, why peptide of digested protein cannot be presented by class 2 glycoproteins? Because the groove is closed by a fragment of the invariant chain called CLIP. This is the right answer. It's most common to present peptide derived from destroyed protein in the cytoplasm or protein which have been eaten by the cell. It's more common to display protein destroyed by in the cytoplasm because class 1 histocompatibility molecules are expressed by all your body cells, while class 2 are only expressed by the specialized cells, normally and after induction. Do you have understood absolutely everything? You want to go through something? Because I don't want to start a new one because it would be too much. I wanted to close with histocompatibility now, and we start with T cells this afternoon.
But is a curiosity just to know, the tumoral cells also present the histocompatibility complex 1, no? The tumoral cells.
The tumoral cell?
Tumoral cells.
Tumoral cell, yes.
Also the histocompatibility complex 1. So
yes,
there isn't a way to, and is a normal histocompatibility complex because there isn't a way to understand if the cells are tumoral or not in this case.
Okay. In general, the tumor have a problem in they elaborated some mechanism to block the presentation of the maturation of histocompatibility molecule. So in many cases, tumor cells are unable to present peptide, are unable to present histocompatibility molecule. Histocompatibility molecules, there is a major rarefaction of this molecule. So there is a mechanism by which normally the tumor cannot be recognized by T lymphocyte. But this is not enough. The tumors induce suppressor mechanism, very strong. So all the area around the tumor is very suppressive. So if a T cell recognizes, which is not common, the histocompatibility molecule, normally is inhibited. And you will see when we go to the control of immune response that nowadays the most common, the most popular way to control the tumor is to break the inhibition. There are some inhibitory checkpoints, it's called PD-1, which the antibody, we try to block this checkpoint and we induce a much stronger recognition of the tumor. But in general, the tumor ability to block the expression of histocompatibility molecule. But since they block the histocompatibility molecule, we elaborated the NK cells which are inhibited by the histocompatibility molecule. So if a cell do not express histocompatibility molecule, it can be attacked very rapidly by NK cell. So the tumor have a problem to control NK cell activity in order to survive.
So the NK cell can be a solution in this case also to reduce the spread.
Well, a lot of people believe that NK cell can be a solution, and they try, as it was in the presentation we saw, there was a company who was trying to exploit NK cell to activate NK cell against the tumor. And some people try to do that. So far has not been very easy. Was possible with certain kind of treatment. We were very toxic, very high doses of IL-2 induce very strong reactivity of NK cells. You can induce tumor destruction, even of very large and diffuse tumor, but many cases the patient die because heart attack and other problem. So the problem of the tumor is very complicated, but if we have time, we will discuss a little bit on this. So you understand the implication of this small group cluster of genes. They are doing something extremely important for our life. They cause our individuality, they signal our individuality, and with T lymphocyte who destroy the foreign histocompatibility molecule, we maintain our individuality. No, a single foreign cell can enter in you without being destroyed by T lymphocyte. So we cannot be contaminated. It's a good, it's a bad sense, because the recognition of foreign histocompatibility antigen trigger very strong reactivity of T cell. And so we have the rejection of kidney, which have been transplanted, of heart, which have been transplanted. And the rejection must be, the transplantation must be accompanied by immunosuppressive mechanism which dampen, which inhibit T cell reactivity. At the same time, having a growth of different kind can allow the presentation of something which other growth cannot allow. In certain cases, we have phenomenon of autoimmunity. The body reacts against its own constituents because some of us inherited a growth which very well presents some kind of certain peptide, or our peptide, and can induce reactivity of lymphocyte. Other person cannot at all have this kind of autoimmunity because they have not the growth to present the peptide. So the diabetes of the young person, the type 1 diabetes, cannot happen in certain kind of person which have certain kind of histocompatibility molecule, because this peptide, which is a target of autoimmunity, cannot at all be present. So to have certain kind of histocompatibility antigen, to have inherited some of this antigen or have inherited other one, is not certainly a neutral situation. A situation in which is very difficult to say is a good combination of the poor combination, because so much depends on the environment. And sometimes the environment is absolutely not predictable, like in the case of HIV, which is a completely new infection, was not existing before, or where some selection were very important when the smallpox in Vaiolo was common in Europe. It was selecting the population. Now this selection makes no sense because smallpox is not existing anymore. Okay, if you have no question, we can stop here and reconvene at 2 o'clock with T lymphocyte. I want to add that. So.